In this video, we will discuss about TGF-β1 interaction with this T-beta receptor 2, and in the case of this double mutant, where they knock down N-glycosylation site in the extracellular region of this T-beta receptor 2, they will check whether this TGF-β1 interact with this double mutant cells or not. So, in order to do this assay, what they did is they used biotinylated TGF-β1 and then used avidin FITC to interact with this biotinylated TGF-β, and we can see the interaction of this TGF-β1 to this T-beta receptor 2 using these FITC signals. So in the case of this wild type transfected cells, we can see that the dose dependent increase in this FITC signal, or that shows the dose dependent increased binding of this TGF-β1 on this T-beta receptor 2. However, for this control is the empty vector transfected cells, or the mock transfected cells, and this dark black is for this double mutant. So when we check the signals for this control and the double mutant, those signals are comparable, indicating that this one of the possibilities could be that this double mutant will not be able to expressed on the cell surface, and because of that only endogenous T-beta receptors to binding occurs. And this is more plausible possibilities based on the results that are presented in this paper, because during the confocal microscopic we have seen that the T-beta receptor 2 in the double mutant case are accumulated in the perinuclear region, mainly in this endoplasmic reticulum. However, the researchers also talks about one more possibilities, that the double mutant are able to express on the cell surface, but they are not able to interact with this TGF-β1 because of the deglycosylated conditions, and the glycosylation plays an important role in this interaction. However, that the chances of that possibilities is lower, because the confocal microscopy shows that the double mutant transfected cells, in double mutant transfected cells, the T-beta receptors are predominantly found in this perinuclear region or in the endoplasmic reticulum. Afterwards, they confirmed the transfection using the Western blot, where they checked the transfected T-beta receptor, transfected TGF-β using the flag tag. Since the control is transfected with the empty vector, they do not see any signals, and this flag tag will show the signals only for the transfected T-beta receptor 2, not for the endogenous T-beta receptor 2. And we can see that for the flag tag we are able to see two bands, and these upper bands could be because of the different glycosylations, different size of glycosylations that occurs. And in the case of double mutant, we are able to see only single band, and the single band also has the lower size compared to the lowest wild type transfected T-beta receptor 2 size. And this could be because of the absence of the N-glycosylation sites, and because of that no glycosylations might occur. Whereas in the case of the wild type transfected cells, different kind of glycosylation occurs at different glycosylation sites, mainly the two glycosylation sites, that is N70 and N94. So this is the final figure of the paper. So from that they conclude that the N-glycosylations of this T-beta receptor 2 plays an essential role for this TGF-β1 interactions, and thereby the downstream signalings. And these things they have confirmed using the dose dependent studies in this figure, and also we have seen in the previous videos, previous results, that they have also confirmed this hypothesis by checking the downstream signaling pathways, activation of downstream signaling pathways using phosphorylated SMAD2. So I hope you understood the story that I presented in this paper. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.